It's my pleasure to introduce Dr. Person, who's at UC Denver in the Physiology and Biophysics department there in the College of Medicine. Abby did her PhD work at the University of Washington with David Pertel in birdsong, and then did 2 postdocs, one in China at the National Institute of Biological Sciences in Beijing, and then she came back and did her work at Northwestern before going to UC Denver. So I don't think I'll take any more time. I'll let her talk. Let's welcome her.
Thanks, Ryan. Allow me to take a moment to figure out how the microphone turns on. Can you guys hear me?
Yeah.
Hear it?
Doesn't sound very loud though.
Let me adjust. Now that I've got it on, angle it. Better? Yeah? Can you hear me? Okay. Just speak up if you can't hear me, and just let me know. Thank you, Simon. Thank you, Bilal, for the invitation to speak at your seminar series. I haven't been to Atlanta since 1996, and it's like I've been plunked into an alternate dimension. It's so beautiful here, and so you should all be very proud of your beautiful campus and city. Today I'd like to tell you about the work that we do in my lab, which is really focused around the topic of precise motor control. And we study mouse reaching movements as a model for this. And we investigate questions of precise motor control at many different levels, studying cells and synapses, circuits in the brain, quantitative characterization of the behavior, so that we can ultimately understand disease models, taking it all the way back full circle to cells and synapses. And so it's, I guess, common these days to make disclosures. I do not have any financial conflicts of interest to report. However, I am going to be talking about the cerebellum. And the reason that requires some amount of disclosure is that apparently it really bores some people, like this illustrator who has fabulously colored the cerebellum in gray. I think that sort of, sells it a little short. The real cerebellum is only slightly less gray. But it's really, in fact, when we start to look at the microstructure and the connectivity patterns within the cerebellum, that its real spectacular beauty is revealed. So this is just a cross-section of a human cerebellum, and you can see what are called the trees of life in Latin. And if you zoom in at a microscopic level, you'll see what Ramon y Cajal really was captivated with, which is a gorgeous microcircuit that's composed of distinct afferents called mossy fibers that contact the supernumerary granule cells, which outnumber all the rest of the cells in your brain. The granule cells project into the cerebellar molecular layer where they contact the spectacular dendritic trees of Purkinje neurons, which are really the workhorses of cerebellar computations. And those send their axons down to the bottom of the cerebellum, which is sadly left down of this drawing in the cerebellar nuclei. And so much of what I'm going to be telling you today is really focused in the cerebellar nuclei, but then at the end we're going to circle back to trying to understand how cerebellar circuits actually compute information that helps movements become precise. Now, I've been alluding to precise movements, and so I'd like to take you back to another contemporary of Ramon y Cajal, Gordon Holmes, who was a physician who operated around the time of the First World War. And he was interested in the neurological deficits that emerged with what turned out to be fairly precise lesions of the cerebellum, in this case, or other lesions of parts of the brain that were produced by gunshot wounds during the war. And what's remarkable about this image is that it's essentially a characterization of a movement that was made in the 1920s. And he did this by attaching a light bulb to the finger of a man with a cerebellar lesion, and then using long photographic exposures so that he could actually characterize the motor deficit that emerged in this particular patient. So what he did is a classic neurological exam for movement disorders. It's called the finger nose to finger task. Essentially, Dr. Holmes was moving his finger to these targets, and the patient was asked to repeatedly point from his nose to these 3 different points in space. And what he observed is that there are these distinct oscillations that occur around the endpoints of these movements. And he thought that these oscillations, which turned out to really characterize cerebellar lesions, were interesting from the point of view of how feedback error-driven control systems work. So as a bunch of engineers, you'll be more familiar than I am with this idea, but it's true, that when you use feedback errors to guide the outputs of control systems, there's typically some level of oscillation that's involved. So the classic example of this is some kind of a thermostat, which uses an error to control the heater in the room, and the room temperature will oscillate around the set point. This is true in reaching movements as well, where if you were to, say, use visual feedback to guide the limb, you would find that your limb careens past its target because visual information is rather slow to get even through the retina. And so the idea is that engineers, as well as the brain, must use some level of prediction to implement feed-forward control so that targets can be achieved without these kinds of oscillatory errors. And because these oscillations emerge with cerebellar damage, it has been hypothesized that some level of feed-forward control is implemented by the cerebellum. So the basic idea is that the cerebellum takes in, through its Mosse fiber pathways, sensory feedback information as well as corollary discharge, and that this information enters the cerebellum through these Mosse fibers, which then are transformed in the granule cell layer to provide rich sensory motor context that can be used by Purkinje cells in the cerebellar cortex to form what are called internal models, or sort of representations and predictions about the body. Purkinje neurons then project into the cerebellar nuclei, including a structure called the interposed nucleus, which are then hypothesized to generate feed-forward control of the body through its outputs to the brainstem and thalamus, which ultimately influence spinal motor networks. So if this is the case, how can we look for evidence of feed-forward control from the cerebellum? So I already showed you that reaching movements are known to be impaired with cerebellar damage, and so those kinds of behaviors are a good place to look. And I also will point out that a number of lines of research have implicated this particular structure, the interposed nucleus, in the control of reaching movements. So this is a study from Claude Getz's lab where they injected mucimol into the anterior interposed nucleus. And they found that reaching movements in cats became somewhat hypometric. And a more recent study from Albert Chen's lab killed output neurons in the anterior interposed nucleus and found that reaching movements actually became somewhat hypermetric in these mice. Now, one of the limitations in sort of understanding the reason that these kinds of reaching errors occur in long-term inactivation studies is that this doesn't really distinguish active control from deficits in initiation or planning, nor does it identify any kind of a control policy. That's really the domain of questions for physiology. And indeed, people have recorded from the anterior interposed nucleus in reaching animals. So this is a study from Peter Van Kaan's group, where they identified neurons in the anterior interposed nucleus that are strongly modulated during reaching movements. However, when the same group had monkeys use a device to push in different directions in the same way that they were reaching in different directions, these cells were very poorly modulated. And so this presented something of a conundrum because, at least in these experiments, they were not able to track the kinematics of the reaching movement when there was a great deal of modulation to study. But when they could track the kinematics, using devices, there was very little modulation to study. So they either had the neural activity and no kinematics, or they had the kinematics and no neural activity. And so this is sort of where the field was, even though there have been other observations relating kinematics to neural activity. There was really no consensus about what the cerebellar output structures encode that's valuable for reaching movements. And so we decided to take advantage of the revolution in motion tracking technology to try to get a better handle on what it is that the anterior interposed nucleus contributes to reaching movements, and started out with just recording neural activity during discrete reaching movements in mice. And so our approach was to use kinematic tracking while recording from the anterior interposed nucleus. So basically, we use an IR-based reflector that allows us to track motions very precisely. So we get real-time kinematic data in 3 dimensions, and this allows us to characterize reaching movements across many, many reaches, so that we get a rich kinematic data set that we can then use to compare neural activity patterns with. So the first thing we observed is that many neurons within the anterior interposed nucleus are modulated during reach. So this is just a raster plot showing neural activity aligned to the start of the reach. And you can see that there's clearly modulation. We can see this among the entire population of neurons that we record, that there's a good deal of neural activity that is occurring during the reaching epoch. But you can also appreciate that this activity is not particularly well structured. And if we return to the observations of Gordon Holmes, as well as the Getz lab and the Chen lab, it seems that maybe aligning to reach start might not be the wisest thing to look at, because there are really preferential deficits in the endpoints of movements rather than anything at the beginning. And so when we align our neural activity instead of to the reach start, if we align to the endpoint, we find that the neural activity is much more clearly structured associated with the endpoint. And this was true across the population, where we find this population of neurons that are very strongly endpoint associated. And you can see that this population is clearly associated with the deceleration of the limb, and as shown here at the population level, endpoint associated. And it really spans the endpoint, so both before and after in this entire outward deceleration epoch. So this is sort of the place where a lot of neuroscience has essentially run aground, is to say, well, we see this neural activity, it's related to the kinematics in the following way, but does this have any sort of causal relationship with, say, producing this endpoint? And so to address that question, we've utilized the fact that we have kinematics in real time to essentially produce a closed-loop modulation of the output of the cerebellar nuclei just during reaching movement. And so the basic idea is that we have a retroreflective marker on the paw. We know where it is in real time. Then we can define a landmark, at which point we deliver a very brief train of stimuli of light to the anterior interposed nucleus that is expressing the light-activated opsin, Chiari-Rhodopsin. The train is very brief. It's just 50 milliseconds long with 2 millisecond pulse widths at 100 hertz. And then we can see whether modulation of the nucleus actually has any effects on kinematics in real time. So here's an example of this experiment. So you'll see in the corner here a little light pop on, or a little dot, in fact. That's just indicating the duration of the stimulation. You can see that the mouse engages in the movement. In this case, it's not successful. Now, there's a lot of reasons why a mouse might not be successful at this task. So it's helpful to look at many of these reaches sort of compiled. So this will illustrate the trajectory of unstimulated reaches and the trajectory of stimulated reaches. These are randomly interspersed along the course of the experiment. so that mouse can't anticipate the stimulation. And so what you'll see is that in these stills, is that initially these stimulated and unstimulated reaches are completely intermixed. But right after the stimulus, you see that the stimulated reaches are lower and not as far out as the unstimulated reaches. And that persists, although there's some late sort of corrective responses by the stimulated reaches. So we can summarize that and show that the endpoints in our stimulated conditions are consistently hypometric compared to the unstimulated reaches. So this provides at least some evidence that activity in the interposed nucleus can actually influence endpoint. But these kinds of positional data aren't as satisfying to look at when we're thinking about the temporal evolution of effects. And so for the rest of the talk, I'm going to be really focusing on velocity curves. So for those of you who are uninitiated to this, I think that you'll be reminded pretty quickly. So this is a very bad PowerPoint illustration of a reach, but the basic idea is that when we reach out to something, there's a very natural bell-shaped velocity curve that occurs. And then when we reach back, there's a bell-shaped velocity curve that crosses zero. And indeed, mice, when they reach, have a nice bell-shaped velocity profile. And now we can look at the effective stimulation on outward velocity and observe the sort of temporal evolution of those changes. Not surprisingly, given the positional effect, we see that there is a rapid decrease in the outward velocity with stimulation, which is shown in blue. And that was true across all of the animals, and it's also true in head-fixed mice. So we don't think this is an off-target effect. We saw velocity effects that were consistent both in the outward and upward direction, such that the vector of change was a consistent decrease in the upward and outward velocity. This we also observed in mice when we restricted opsin expression to premotor output neurons that project to the red nucleus. And so this shows that interposed neuron excitation alters reach kinematics in real time in a reproducible directional vector. So this raises the question of whether this might reveal a mechanism for precise cerebellar control of kinematics. So to begin to address that question, we took advantage of the fact that we can alter the power of our light, which alters the amount of spikes that we add to the system. And we can also alter the position of our stimulation. So when we stimulate either early, mid, or late in the reach, we always see the same direction of change. And similarly, when we vary the power of our light, we find that we can vary the magnitude of the velocity change. So this suggests that when we look at neural activity in the endogenous state, that perhaps neural activity is dictating when and how fast the limb needs to decelerate. So the next thing that we wanted to investigate was, instead of just introducing this kind of perturbation, we wanted to take away spikes to say, is the endogenous mechanism that we can record in vivo producing some causal control of limb kinematics? And so to ask this question, we used essentially the identical method, except now instead of exciting neural activity, we use an opsin that can turn down neural activity. And that's called ARCH in this case, that we used an archirhodopsin that is a proton pump that can hyperpolarize neurons. And we can show that this very effectively hyperpolarizes neurons. So what happens when we do this during reach? So when we stimulate during the reach, we're actually able to accelerate the limb wherever we put our light pulses in. And we can see that clearly in this difference plot, where we increase the outward and upward velocity. And so you can see that that vector of change is in the opposite direction as the Chandler-Rhodopsin stimulation. So we're accelerating the limb as if we're removing some kind of a brake from the system, and that ultimately produces significantly hypermetric endpoints. Now, I just alluded to this idea that maybe there's a break in the system and that we have big breaks, little breaks, smaller breaks, or the brake is turned off, right? But this set of findings is essentially consistent with two different models. So just to orient you to how we're thinking about this, we know that motor cortex is essential for producing reaching movement. Strokes cause debilitating effects on reaches. You can't reach anymore. And optogenetic manipulations, as well as mucimal inactivations of motor cortex, abolish reaching. So what we think is that motor cortex commands reaching movements, but it does so through innervation of a whole variety of brainstem structures. And so since we see that there are these effects by the cerebellum, what is it doing to these motor cortical commands? So what I've shown you so far is that we know that when we stimulate, that we reduce outward and upward velocity of a reach. So that could be a brake, suggesting that we're just sort of scaling this control motor signal by some amount less than 1. And that would also be, the observations would also be consistent with producing some kind of a directional command, so that we have a vector of change that's in this direction that we're adding on to our control vector. Now, thus far, our data do not distinguish these models. However, we would make a prediction that if we were to stimulate upon return, that these two possibilities would produce differing predictions. So with our brake model, we would predict that if we stimulated on control, we would slow the return of the reach. However, with the directional command model, we would accelerate the return of the reach, because we'd be adding on this vector right along the top of the control movement. And so we performed this experiment, and the data are consistent with this directional command model. So essentially, when we stimulate during return, we accelerate the limb back to the body. And so we think that essentially the output of the cerebellum is added onto the cortical commands that are descending into the brainstem, and added into it in a directionally consistent way. So these ideas that we're revealing with optogenetics prompted us to ask kind of our original question again of the neural data, which is, what are the principles of feed-forward control by the interposed nucleus? Is there something deeper that we can say than, well, there's a directional command that's added on to the motor cortical signals? And so we went back into our data, our neural data that we had recorded in the nuclei, and we said, okay, so what is this actually encoding? So as you can see, there are trial-by-trial variability in the output of this cell. And we can, in fact, perform simple correlational analyses and show that the firing rates are related to the amplitude of the deceleration of the limb. And that was true in a large number of the endpoint-related cells. So we get this linear relationship. And then we ask, well, then why is it variable from reach to reach? And so to address that question, we grouped reaches by firing rates, which are indicated in this color map. So these are the lowest firing rate reaches, and these are the highest firing rate reaches. And consistent with the linear correlations, we find that the highest firing rate reaches have the fastest deceleration, and the lowest firing rate reaches have the slowest deceleration. So, but why? So what was interesting is that if we went back in time from this period in which we're analyzing neural firing rates, we found that the fastest decelerations happen to occur in reaches that had the highest peak velocities, as shown here. So, so this suggests to us that the fastest reaches had to decelerate the fastest, and indeed that was produced by this variable change in the output from the cerebellum. And I think it may be clear or obvious that if you have a very fast reach and you decelerate faster, that you're going to produce stable endpoints. And that's exactly what we observe. So this was interesting, and we looked at it carefully along these along the range of firing rates produced by the cerebellar nuclei, and we found that there was this very nice matching of the amplitude of the peak velocity and the amplitude of the deceleration, such that the fastest reaches had the fastest decelerations. Note that this is an inverted scale. And so there was this tight relationship between peak velocity and deceleration that was produced by this variable output in the cerebellar nuclei. Now, just to emphasize sort of the relationship between this idea and stable endpoints, we produced a very simple model in which we applied the average deceleration to reaches that were the slowest. And when we do that, we find that if you decelerate too fast on a slow reach, you end up with a hypometric reach. And similarly, if you have an average deceleration on the fastest reaches, you end up with a hypermetric reach. And so it's really this variable tuner that's happening in the interposed nucleus that we think produces this tighter range of endpoints. And so this observation of adaptive scaling by the anterior interposed nucleus is, we think, evidence for feed-forward control by the cerebellum, that it is an online modulation of reach kinematics that's based on the absolute value of the reach kinematics beforehand. So that we get these this kind of adaptive scaling. And so this raises the question of how is that computation actually made? And so for the next several minutes, I'm going to be telling you a little bit about our work in identifying or getting a grip on how these feed-forward computations are made. And so we will start with the premise that feed-forward control and computations made by the interposed nucleus are in part going to be derived by the inputs to the nucleus. And so I have been talking about this as sort of a monolithic structure for a little while, but in fact, the interposed nucleus is composed of multiple cell types. And among these are output neurons that project to premotor targets, like the red nucleus and the ventrolateral thalamus, among other places, as well as an inhibitory neuronal population that projects to the inferior olive, which is another structure that I won't be talking much about. And what we wanted to ask is, what are the inputs to these premotor targets, since we know that these are sufficient to modulate reach kinematics? So to address this question, we used a method that was developed by Ed Calloway's lab at the Salk Institute, where you're able to introduce a rabies virus to specific cell types driven by a Cre promoter. The rabies has been modified so that it can't actually jump synapses and infect the whole brain, because a glycoprotein that's required for synaptic jumps has been left out of it. But in order to get one synaptic jump, the idea is that you introduce that glycoprotein just to Cre-expressing cells. And so in trans, the glycoprotein can be complemented with the rabies virus, and then it can jump. However, the rabies virus is stuck in the next cell, and it won't continue to infect the brain because it doesn't actually express encode the glycoprotein itself. And so we have identified a Cre line that expresses in premotor output neurons of the cerebellar nuclei called NTSR1. These neurons are not the same as the GABAergic neurons that I said project other places. And so we've used actually both of these lines, an NTSR1 Cre line and a GAD1 Cre line, in combination with these viruses that ultimately allow us to analyze the inputs to these different cell types. And so we perform these experiments, and we can map inputs to these different cell types. We can analyze the location of all the Purkinje neurons that go to these cells. But more importantly for this experiment, we can analyze the location of all the extracellular inputs that go to these targets and say, well, what kind of information is probably giving rise to this adaptive scaling? Obviously, I don't expect you to read all these labels, but what I want you to appreciate is just the colors of these bars and the relative colors of these bars. And what we consistently observe is that structures that are considered motor structures are by far the dominant inputs to the cells in the cerebellar nuclei, more so than sensory structures. And so if we think about this adaptive scaling that we observe in the cerebellar nuclei, we might think that perhaps some of that is derived from the fact that the whole motor system is turned up and down with the amount of motor vigor, and that the nuclei might inherit some of that information directly. But that presumes that the Purkinje neurons aren't also modulated in concert. And so we were interested in this idea, and we turned to a motor structure called the red nucleus to better understand this idea that motor information, which is also called corollary discharge, might enter the cerebellum through these mossy fibers. And we consistently observed retrograde label in the RNM from these rabies experiments, and so we chose to just inject a standard anterograde tracer into into the red nucleus. And what we observed was really striking to a cerebellologist, and should be striking to you too in a second. So we observed that there was very dense innervation of the anterior interpose from the red nucleus. However, what we found striking is that there was almost no innervation of the cerebellar cortex. And that's unusual because a typical mossy fiber pathway lights up the cerebellar cortex with countless what are called mossy fiber rosettes. These are the endings of the mossy fibers. And so those were not there from the red nucleus. And this suggests something of an alteration of this circuit motif that we draw all the time, which is the idea that mossy fibers enter the cerebellum and innervate both the cerebellar nuclei and the cerebellar cortex through these mossy fibers. Instead, we observe that there are at least some sources of mossy fibers from motor structures in particular that only innervate the interpose nucleus. And so that's further evidence that there can be sort of an independent modulation of cerebellar output that might scale directly with motor vigor to produce scaled deceleration. Now, this sort of leaves the Purkinje neurons out and the whole cerebellar cortex out. And we don't think that they're probably doing nothing. So in the one remaining minute, I'm just going to talk about how we are thinking about what the cerebellar cortex might do. So I've given you this model now a bunch of times where we have sensory information and motor information coming into the cerebellar cortex and ultimately producing feed-forward control. The idea here needs to be a little bit spelled out in that the mossy fiber information is thought to be a high bandwidth signal. So say any kind of information about velocity would be continuously encoded by mossy fibers. And there would be this huge amount of information coming in. But this kind of information is hard to learn on, it's thought. And so it's hypothesized that this context layer, which is the granule cell layer, decomposes these signals into tiny little basis sets. And that these little signals coming out of the granule cells are more conducive to learning by the Purkinje neurons than otherwise would occur. And so I'm not going to go into cerebellar learning theory, except to say that we think that there's probably some kind of learning that happens at this level that probably contributes to feed-forward limb control. So to address that, we've begun recording from Purkinje neurons in head fixed mice that are reaching for a food pellet. So when we record from Purkinje neurons, similar to the nuclei, we notice that there are many neurons that are reach associated. And what we ultimately want to know is whether this kind of activity can help produce stable endpoints. And so what we have begun to do is stimulate the mossy fibers that enter the system. But we're doing this in a slightly different way than we're doing the stimulation optogenetically in the interposed. So here we're using optogenetics, but we're doing this in blocks of trial rather than randomly interspersed. So the mouse is able to reach for the target 25 times, and then we begin a block of stimulation where every reach we stimulate at the same landmark for 50 reaches, and then we take the stimulation off. And the idea is that this stimulation, because it's occurring at the same landmark every time, should become a predictable signal that the Purkinje neurons could use to help guide the motion. And so what we observe with respect to endpoints is that we have a spread of endpoints before we start the stimulus block. Then we start stimulating on every trial, and we're able to actually push the endpoint around. However, we notice that indeed, as we predicted, that the endpoint actually begins to shift to become eumetric to the original endpoints. And that when we turn the stimulation off, that there is a predictable rebound such that the reaching endpoints are hypermetric briefly. And so together, these data suggest that indeed there is probably some kind of an adaptation that's going on in Purkinje neurons to ultimately guide endpoints in the produced by the interposed nucleus. But the work analyzing the neural effects of these perturbations is ongoing. So that's where I'd like to end, and I'll take questions afterwards. But the basic idea that I want to emphasize is that we've identified a feed-forward control policy from the cerebellar output nucleus, the interposed nucleus. And that policy is essentially to adaptively scale the deceleration of the limb through variable firing rates. And we've made some progress in identifying how motor signals from the brain may enter the cerebellar nuclei directly, and that afferents coming into the cerebellar cortex really can be scaled such that they control endpoints in bidirectional ways. And so you'll have to stay tuned until we can actually tell you more about how those Purkinje signals are produced. But I'd like to acknowledge the very hard work of the students that I have had the privilege of working with. Matt Becker did all of the work in the anterior interposed nucleus. Dylan Collin is working on the Purkinje cells. Elena Judd is my technician who's done a lot of the rabies tracing and analysis. And Kristi Beitzel did the work in the red nucleus. And so I think we have plenty of time for questions. So if you would like to let me know if you have any questions. Thank you for your attention. Peter?
Yeah, thank you. That was very clear and so straightforward that it almost applies to cerebellum messages. So the last set of results reminded me of Tom Thatcher's experiments with throwing darts at the Francis goggles. It seems to be sort of the same kind of time course. And so would you say that the throwing darts circuit is the same as the one you're describing? And the visual input is the context by which you can shift around the muscle fibers?
Okay, so Dieter is describing this classical cerebellar adaptation experiment in which you can throw darts at a target, and you're on target, and then you put on prism goggles, which distort your visual field by some number of degrees. And then, you start throwing off target. You think you're throwing forward, but you're really off. And then quickly you adapt and you're now on target. You take the goggles off, but now you overcorrect in the opposite direction. And it's found that in cerebellar patients that they cannot produce that adaptation. And so first of all, yes, I think this is exactly in line with that whole model. And what's interesting about this is that this particular version of this is that we're producing that distortion with mossy fiber stimulation itself, and the cerebellum seems to be able to adapt. So that isolates it again to the cerebellum, which is good news for us in studying the Purkinje fibers. But I think that what's, unclear in all of these experiments is what is the information that's being used for the Purkinje neurons to make their computation. And so in all of these cases, we have cortical input through the pontine nuclei. That's undoubtedly important. Then we have all of this feedback from the brain stem that's coming in through mossy fibers and the spinal cord. And so isolating, which pathway is doing this and which pathway is doing that is almost intractable, I think, under sort of endogenous conditions. And so our hope using this model is that we can just basically hijack a mossy fiber system. We don't know which one it is. We don't necessarily care. And we can produce the contextual information that's either going to be helpful or hurtful to the system, and then test whether it actually is usable. So we think it would be exactly the same mechanism, and it's probably the same mechanism that's going on in other parts of the cerebellum. So does that answer your question? Yeah. Yeah.
I have a question. So I really like the perturbation sort of aspect of the experiments you're doing, and it reminded me of the more exogenous perturbations people use with manipulandums and in human reach or monkey reach and so on. But I started thinking, okay, but in that case, many times the perturbation is, pushing them off a certain trajectory, maybe orthogonal to the path,
right.
As opposed to, it seemed like what you were showing was the perturbation was always along a trajectory, either decelerating or accelerating along a predefined trajectory.
Right.
Is there are there ways to push it off the trajectory?
Okay, so I think your question is like the root of a whole set of experiments that we're trying to address, because we've been wondering, why is it along this trajectory? So I'll get to that in a second. So first of all, we have wanted to do some exogenous perturbations in a variety of directions, and have spent a certain amount of time developing these kooky, like closed loop puffs and electromagnets that can pull the limbs so that we can do these kinds of adaptation experiments. I'll spare you all the gory details because they haven't worked that well. But the question remains, a good one is why is this, why are the direction of changes so sort of natural? And so first of all, we don't know whether the cerebellum is encoding something in sort of a body-centric coordinate frame, which would possibly, what we're calling a vector of change in this direction is really a general vector back to the center of the body. And so we're doing experiments now to alter the position of the target, and we would expect that depending on the axis of movement, that some of these directional changes that we found to be invariant with one target are actually variable. So for instance, if the animal's reaching down, we might predict that it's actually up and back rather than down and back. Okay. So those experiments are ongoing. But in theory, the system, depending on what its overall sort of coordinate of control is, could handle orthogonal perturbations in the same way that it's handling any of these directions. But we have to have a grip on what that coordinate frame is first before we can start to make clearer predictions about how it should respond if if it's pushed over this way or that. Does that answer your question? Yeah, Simon.
There was the old, I think along the line of what Garrett was asking about, like moving in force fields and such. And I'm just wondering if clearly kinematics matters a lot because the endpoint, they want to reach for that. But is there a representation when they're building a feed-forward model? I guess my question is, how are they incorporating things like inertia of the limb and stuff like that? Is that taken into account at that stage in building that feed-forward model? So if you suddenly make the limb heavier, but not necessarily deflected off of its trajectory, such that the same feed-forward command would now be erroneous, but
That's
wouldn't
right.
produce an error automatically.
Yeah, that is exactly what cerebellum is predicted to care
Right.
about. So all of that, the physics of the environment and the physics of the body are presumably learned by the cerebellum so that it can make these predictions. And really, when you watch babies develop and things like that, I recognize there's other things going on, but a lot of that is probably training the internal model to be able to handle these, complex physical interactions. And so you see a lot of play that's like highly bizarre.
But
Why
so
are you
then
standing on your head constantly?
you would need to, so you're saying you would need to adapt that feed-forward model over many trials then,
Yes,
in order
that's
to
right.
deal with that. We couldn't deal with a transient perturbation. in
Well,
inertia.
I think the idea is that a transient perturbation happens and there's rapid correction from that. But there's good evidence to suggest that a lot of that is cortically mediated. So the idea, though, is that the cerebellum cares about that deviation from prediction, and then over trials would retrain that. And so then you get adaptation over trials. And then if that perturbation is now gone, there has to be re-adaptation.
So I guess my critical question then is, to make it more concrete, is it learning, is it known that it's actually learning the physics, as you say, or learning the typical kinematic deviation that physics produces?
Yeah, I mean, maybe we'd have to talk more deeply about the differences there. I mean, what we can say is that in experiments in which people have been able to isolate specific sensory command or sensory stimuli, Purkinje neurons can disambiguate like vestibular signals that are equivalent to gravitational pulls. And it's thought that it's really through combinations of inputs that are like hard to replicate that, like, for example, if there's a vestibular illusion of falling, that's the same as some kind of linear acceleration in a different direction. The system can disambiguate that through using combinatorial codes. And so I think maybe the answer is that it's not solving a physical problem in the same way that a physicist would, but it's able to use these kinds of signals in ways that are ultimately helpful for the system to operate. But I think it's maybe too complicated to answer right now.
Okay. I think we should wrap up then. So let's thank Abby one more time.